Hello, everyone. Welcome back once again. Today we are going to discuss another topic from prokaryotic transcription. It is in continuation with the last topic of initiation. It's elongation and termination. So, so far we have studied that in case of prokaryotic transcription during the initiation, RNA polymerase, with the help of signal factor, it starts as a closed complex bind to the promoter region. Then it transitions into an open binary complex by the process of isomerization. And ultimately, um, once the polymerase become active, it forms a stable ternary complex, which will include the RNA polymerase enzyme, DNA template, as well as the RNA transcript. We also have studied in the previous lecture that there is a process of abortive initiation, which only after which the process of transcription can leave the initiation phase and enter into elongation phase. So that abortive initiation, it also um, ends up in the escape from the promoter, meaning the promoter site, it is transcribed and now RNA polymerase is going to move forward from the promoter region. So that marks the elongation process. Now, what will happen when the elongation process starts? Now, the RNA polymerase, it is still the same polymerase, but now we call it as an elongating polymerase because of its changed processivity, as well as at the same time, it is also doing a proofreading activity. Processivity meaning the rate of polymerization has increased because now the polymerase, with the help of the beta pincers, they are tightly bound to the DNA template strand, and they are moving forward from 5' to 3' and polymerizing the ribonucleotides one at a time in its active site. Let's see the details about the elongation phase. So this slide, it shows us the details about the elongation phase. And here, the first step which you can see is the physical process. Physical process meaning what actually is happening with respect to the entry and exit of DNA, RNA template, where the RNA DNA hybrid is forming, all those minor details with respect to the location of the all these things. So here, if you see, this is a RNA polymerase enzyme. So it has this one channel which is called as the ribonucleoside triphosphate uptake channel, so marked by A. So this channel, with in this channel, continuously these RNTPs they are coming through, and then we have in the inside of the RNA polymerase, we have the active site where these RNTPs, they will actually form a complementary base pairing with the template DNA, and then the phosphodiester bond will be formed between the two consecutive RNTPs. So when they are forming the bond and they are reading this with the help of the polymerase, they are reading the nucleotide bases on the DNA template. So only about 8 to 9 base pairs, if you see, there is 8 to 9 base pairs space in the polymerase where the DNA RNA hybrid can be formed. And after that, once it's formed, the RNA has to exit through the RNA exit channel, and the DNA, both template and non template strand, will leave from their respective channels. So that's how the physical process works during elongation. So from one end, the double helix from the pincer ends, the double helix is going through, the unwinding is happening simultaneously, and then DNTPs are coming. They are forming the phosphodiester bond, and RNA transcript is leaving the RNA polymerase. Likewise, these two, no template and non-template, will again leaving from their respective exit and forming the double bond outside the RNA polymerase. So this is physical process that happens during the process of elongation. again, now if we look at the chemical side of the, what's the chemical story behind this? So chemical story meaning on this, in this active site, what is happening, how the RNTPs, they are coming and they are forming the phosphodiester bond, what kind of reactions happen. So we'll see that in the coming slide. But before that, let's see what are the raw materials. I think from very earlier classes we know that how the DNA and RNA, what are the building blocks. So here the RNA building blocks are the RNTPs, which is ribose sugar. Then we have a nitrogenous base, and then we have a triphosphate group which provides the energy. So here the ribose sugar, it forms a very crucial role in forming the backbone of the any type of nucleic acid, whether DNA or RNA. So the phosphodiester bond, it is formed with the help of this 5 and 3 locations. So if you see the numbers, the positions on this ribose, so we have this one where the N2 bases are attached always, so it's always already not available. Then we have 2, then we have 3, then we have 4, and then this one is the 5. So this, with this 5CH2, it's there is the triphosphate group attached. Now, with the help of the 5 dash and the 3 dash ends of the consecutive RNTPs, the uh, chemical process will happen and a phosphodiester bond is formed between 5 dash and 3 dash positions. We will see in detail in this particular image. So here, if you see, this is a condensation reaction where we have where we have this 2 and 2 RNTPs actually. So here there is, this one is the if you see, it doesn't have additional, um, p. So it just has a one p because it's involved in the phosphodiester bond formation. So long chain is not shown here, but it reflects that. So this is the nitrogenous base, or this is the ribose sugar that's present in the already existing RNA transcript, and then a new RNTP is coming from the RNTP uptake channel. So here we have this nitrogenous base, this ribose sugar, and a triphosphate. So this triphosphate attached to this 5 dash carbon, 5 carbon. Okay. Now what happens here? The hydroxyl group, which is present on the third position, it will perform a nucleophilic attack on this first phosphate, releasing this pyrophosphate. Inorganic pyrophosphate is released, a water molecule is released, and this result into the phosphodiester bond formation between the 5 dash 3 dash location. So that's how the chemical process happen, and the energy, it's coming from the pyrophosphates, and the process is catalyzed with the help of the RNA polymerase. So this is something that's happening within the RNA polymerase in its, on its active site. So, and we also know that DNA, from DNA polymerase, RNA polymerase is different because it can catalyze this reaction. It can perform this condensation reaction without any existing strand, meaning it can polymerize the RNTPs starting from the raw material. Okay. Now this is the chemical process. Let's move on to the another aspect of this elongating polymerase. So now the polymerization is happening and the transcript, RNA transcript, it's keep on growing. But we know that there are some chances of the errors in them. So if there is a mismatch, then that mismatch can create an error in the mRNA transcript. So polymerases, they have a capability of both the polymerization activity, and then we have also the endonuclease activity on its one end. So this endonuclease activity is basically responsible for the process of proofreading. So here again, the RNA polymerase proofreads also the RNA transcript, which it is producing, and it's doing it in 2 different ways. The proofreading process will be done in 2 different ways. We'll see, and that is done with the help of backtracking. So what happens whenever the RNA polymerase is moving forward, it identifies that a mismatch happened in its previous step, could be just one step back or could be few steps back. Then it will identify the mismatch and it will come back to that mismatch, try to remove it and replace it with the correct matching base. How that happens? So, whenever RNA polymerase it's moving forward and catalyzing new RNTPs into the RNA transcript, if there is a mismatch, अगर कोई भी एंटो बेस जो है वो गलत गलत जगह पे गलत पोजीशन पे आ गया है, वो क्या करता है, वो जो आपका ट्रांसक्रिप्ट है या डीएनए आरएनए हाइब्रिड है, उसकी कॉन्फर्मेशन को वो चेंज कर देता है, यानी कि उसका जो स्ट्रक्चर होता है जैसा होना चाहिए था वैसा नहीं रहता। और जब वो स्ट्रक्चर मतलब मिसफोल्ड हो जाता है या फिर वो एक डिफॉर्मिटी आ जाती है, उस डिफॉर्मिटी की वजह से क्या होता है, दैट मिसमैच, it becomes exposed to the endonuclease site of the polymerase enzyme. The same thing happens with the DNA replication or DNA polymerase enzyme also. So here, so now what happens once it identifies that there is a mismatch, it can do two things. One, it can do pyrophospholytic editing or it can do hydrolytic editing. What is the difference between the two? So this image, it is pyrophospholytic editing, and this image it shows the hydrolytic editing. Now, and both are catalyzed by the endonuclease active site of the RNA polymerase. So in this particular first case where the pyrophospholytic editing happens, here the polymerase enzyme will identify a mismatch just one step back. So it can catalyze one step back reaction. And what it will do once it moves on to the polymerization of the next nucleotide, it will identify to come back. Now this when the new uh, uh, polymerization, when the new RNTP is being added, we have this available pyrophosphate. So once it come back, it utilizes this pyrophosphate and reincorporate in this particular N2 base and removes it. And once it removes it, then it comes back and catalyze the correct nucleotide bases. That's how the pyrophospholytic editing will happen. So reincorporation of pyrophosphate and it corrects one step back reactions only. Now, what will happen in case of hydrolytic editing? Hydrolytic editing में क्या होगा कि let's say जो आपका polymerase है, it moves few steps away from the mismatch, but it identifies that there is a conformation change or there is a structural change happened at some point a little bit away or back. Then it will come back to this location, come back few steps back, and then it will remove all those nucleotides. So it will break down here and it will start performing the or correcting the nucleotides right from the mismatch. So with the process of the condensation reaction, तो वो polymerization जो है cleavage करके निकाल देगा, and then polymerase activity will take over and it keeps on adding the now correct matching base pair. So that's how the two different processes of proofreading happen during the elongation phase. Now, at the end of this, we have the termination. So, termination is the final step in the prokaryotic transcription. And termination, it is marked by the presence of certain termination sequences. Now, in case of this prokaryotes, termination, it is of two types. One is row dependent and another one is row independent. Now, what is row? Row is protein. It's a factor, protein factor, which identifies certain set of sequences and helps the RNA polymerase to know that now the process needs to be terminated. However, in case of row independent mechanism, there are certain other factors which we are going to just quickly see. So in case of row independent, no protein factors are involved. So there is no row as such. But what happens then? So there, in addition to other than that, they have this two sequence elements of short inverted nature followed by 80 pairs in double-stranded DNA. What does it mean? So here, if you see that we have two set of sequences, so one this side and the other one is this side. And these sequences, they are inverted. So inverted meaning A, G, C, 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 G, and then the same thing will be repeated in this order. So G, C, G, G, T. So this is like they are present but in an inverted manner. And because of this inverted arrangement of the sequences, these sequences, once they are transcribed, they will have a tendency to form a pairing. ओके, सो वो क्या करते हैं, क्योंकि यहाँ पे ये जो सीक्वेंसेस एक दूसरे के कॉम्प्लीमेंट्री हो जाते हैं, जब ये ट्रांसक्रिप्ट फॉर्म करते हैं, दे create a hairpin like structure. And once they create a hairpin like structure, and it is followed by what? It is followed by AT base pairs. AT base pairs जो होते हैं इसके आगे हैं, यानी यहाँ देखो अगर आप, तो टी 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 टी, ये सारे के सारे जो हैं वो आपके टी हैं। और जब ये ऑलरेडी जो है इनमें रिड्यूस्ड वीक स्ट्रेंथ होती है इनकी, और जैसे ही ये हेयर पिन लूप बनता है, तो पॉलीमरेज एंजाइम जो है वो थोड़ा वहां यहाँ पे स्टक हो जाता है। एंड बिकॉज ऑफ द प्रेजेंस ऑफ टू लाइक structure which is sequences which are of weak strength, it falls off from that particular region. So this is how it looks like. So the RNA transcript, it keeps on producing, and once it identifies these, it produces this sequence, like this one. In this case here, the sequences are produced and it forms a hairpin loop or hairpin like structure. So U, so this region will be what? It will be of weak strength. And because of this weak strength in nature, when RNA polymerase starts going to this direction, this and weak strength, it acts as a hindrance or obstruction, which helps in the release of the RNA polymerase from the DNA template. And that's how the process of replication it will end. Now, another one is the row dependent. Row dependent mechanism क्या है? उसमें एक आपका प्रोटीन फैक्टर है रो, जिस जिस जो क्या से बना हुआ है 6 हेक्सामेरिक प्रोटीन्स के साथ, और वो एक रिंग की तरह स्ट्रक्चर बनाता है which is open at one end. So here in this mechanism, what happens? The row factor, it has a capability to identify RUT sequences. RUT sequences जो होते हैं वो कहाँ पे प्रेजेंट होंगे? आरएनए ट्रांसक्रिप्ट के ऊपर। तो जब डीएनए को आरएनए पॉलीमरेज ट्रांसक्राइब कर रहा है, तो आरएनए ट्रांसक्रिप्ट में कुछ सेट ऑफ सीक्वेंस होंगे जिसको हम बोलेंगे RUT सीक्वेंस, which is nothing but row utilization sequences. And these sequences, once row identifies it, it will come and bind to it with the help of ATP. And what will happen? This transcript will um, weave through all these मोनोमर्स। ये आपके जो मोनोमर्स है जो हेक्सामेरिक रिंग बना रहे हैं, उनके अंदर से ये वीव हो जाते हैं थ्रेड की तरह। एंड एट in from the center, the center is around 12 angstrom. From this center, it leaves the zone. And once this conformation is formed, it result into the immediate release of the RNA polymerase. So ये एक तरीके का नॉट है जो रो की हेल्प से, RUT सीक्वेंसेस की हेल्प से RNA में प्रोड्यूस होता है, एंड दैट रिजल्ट इनटू द termination of the process of transcription. I hope this is clear. Process of transcription prokaryotes के केस में initiation in one video and elongation and termination in this particular video. If you guys have any questions, feel free to reach out to me. And with that, we are going. Thank you. Bye bye.